This is part 2 of 2 on chapter 2, the chemistry of biology. In this part of the chapter, we're going to be looking at something called organic chemistry. So we're going to be focusing on carbon, all the different types of molecules that are carbon-based, what those functions of the molecules are, and how they relate to cells, which we'll get to in the next unit. So in organic chemistry, we're going to specifically be looking at carbon. So organic molecules are just compounds that contain carbon bonded to hydrogen atoms. So carbon is the fundamental element of life. So if you look on the periodic table, carbon has an atomic number of 6, which means it has 6 protons. It also has an atomic mass of 12.01. We're just going to go with atomic mass of 12. So this means that you have 6 protons and 6 neutrons, so that gives you atomic mass of 12. And you also are going to have 6 electrons. So 2 of those electrons go into the first shell, and then that first shell is full. So then we have 4 more electrons, and those 4 electrons go into the second shell. And you can see that in the picture on this slide up here. So those 4 electrons that are in the second shell, or the outer orbital, those 4 electrons can form different types of bonds with other atoms. So carbon is very versatile in how it can bond. So if you look on the left side of this slide, you can see that carbon can bond with lots of different types of atoms. So up at the top we have carbon bonding with hydrogen, carbon with oxygen, carbon with nitrogen, carbon with other carbons. You can have single bonds, you can have double bonds. At the very bottom you can have carbon and nitrogen, and it forms a triple bond. So it can, lots of different atoms, different types of bonding. In addition to bonding with different types of atoms, you can also have linear bonding, so just a straight chain of carbons. The carbon chain can be branched, can be very highly branched, as we'll see in some examples. You can also form carbon rings, and we'll see these carbon rings in certain structures. So carbon is that backbone of life, and it's very, very versatile. It can do lots of different things. So those different atoms that can bond to carbon, sometimes that atom will actually be like a functional group. So it's a group of molecules, they're accessory molecules, that bind on to your carbon chain or carbon ring. And these functional groups, they have unique reactive properties for that whole molecule. And we have a table here showing different functional groups. You don't have to memorize the names of the functional groups. Just be aware that there are functional groups that are found in certain types of molecules. So up at the top we have a hydroxyl functional group. It's an OH. Those are found in alcohols and carbohydrates. If you go down, you'll see there's an amino functional group. Those are usually found in proteins, they're found in nucleic acids, and then you can also see other types of functional groups. So you're going to see some of these show up when we start looking at the different biological molecules. So we're going to be looking at these biological molecules. There's actually 4 different types we're going to be looking at. These biological molecules are called macromolecules. So they're just really large compounds or really large molecules that are put together from smaller subunits. And these smaller subunits are usually repeating, so they're called monomers. So you're going to have these building blocks or these repeating units called monomers. We're going to put them together to form a polymer, which is just many of these repeating subunits. So it's just a chain of monomers that have been put together. So you can see that in this reaction at the bottom of the slide. So we have 2 monomers as our reactants. They go through a chemical reaction. It's a synthesis reaction. So you're going to put the 2 monomers together to form a polymer. So now there's 2 monomers bonded together. So the 4 different types of biological molecules we're going to look at are carbohydrates, lipids, proteins, and nucleic acids. So we'll start with the carbohydrates first. The carbohydrates, their monomer is called a sugar or a saccharide. So those are the building blocks. And these saccharides, they can be 5 carbon rings, they can be 6 carbon rings. So these carbon rings, they form, or they're going to bond together to create polysaccharides. So that's many of these saccharides, or many of these monomers hooked together. So examples of 6 carbon monomers include glucose. We also have galactose. And then fructose is another example over here. We're also going to have deoxyribose, ribose sugar. So there's lots of different types of sugars that can be hooked together to make these larger carbohydrates. So these saccharides are the simple carbohydrates. If you have one saccharide, it's called a monosaccharide. So mono means one saccharide. And they're anywhere from 3 to 7 carbons. Usually 5 or 6 carbon rings make up a monosaccharide. If you join 2 of these monosaccharides together, you're going to get a disaccharide. So di is 2. So 2 of these joined together. Anything over that we call a polysaccharide. So they define it as 5 or more of these saccharides joined together, but it can be anywhere from, more than 2. You can consider that to be a polysaccharide. In order to get a disaccharide or polysaccharide, we need to join 2 saccharides or 2 or more saccharides together. So these subunits, these monomers, or these saccharides, we're going to bind them together using a dehydration synthesis. So basically we're going to remove a water molecule, and that's going to allow these 2 saccharides to join together. And the bond that forms between the 2 saccharides is called a glycosidic bond. So if you look at the image in the middle, where we have 2 glucose molecules, those 2 glucose molecules go through a dehydration reaction. So we get a molecule of water that's taken out. And then you get a bond that's going to connect your 2 rings, and that's your glycosidic bond. So you have 2 glucose molecules, they come together, they form a disaccharide called maltose, and we get water as a byproduct from it. The function of carbohydrates, they function for cell structure. So we're going to see carbohydrates help to make up the plasma membrane, they help to make up the cell wall. They're used in adhesion, so helping cells stick together, either 2 cells sticking together or a cell sticking to some other type of surface. And then finally, carbohydrates are also used in metabolism. They're usually used for short-term energy storage. Our second type of biological molecule we're going to look at are the lipids. So lipids, they're really long, complex molecules. The main defining feature is that they're hydrophobic, so they're water-fearing. So lipids, you're going to see them have a lot of non-polar covalent bonding, which makes them hydrophobic. So it's a lot of carbon-hydrogen chains hooked together. So there's 3 different types of lipids, and they each have their own specific function. So you have triglycerides. These are like the fats and oils. They're used for long-term energy storage, so think of storing fat. The second type of lipid is called a phospholipid, which I mentioned in the previous part of this chapter. Phospholipids are one of those amphipathic molecules that have both a hydrophobic and a hydrophilic part to them. So these phospholipids are going to make up the majority of the cell membrane. And then our third type of lipid are the steroids or hormones. These are found in cell membranes, so usually we see them as cholesterol. So they help make up cell membranes, they help to regulate how fluid that membrane is going to be for the cell. So the triglycerides, they're made up of 3 fatty acids, so 3 carbon-hydrogen chains, and those 3 fatty acids are bound to a glycerol molecule. So like I mentioned, triglycerides are used for energy storage. They can either be saturated or unsaturated. Saturated fatty acids are when the carbon chain is straight. completely straight. And these saturated fatty acids are solid at room temperature, so they're usually found as animal fats. The other type of fatty acid is called unsaturated. This is where not all of the carbons are bound to hydrogens. Some carbons are double bonded to other carbons. And this causes the chain to actually have a bend in it, as you can see at the bottom right-hand side of the slide. Because these fatty acid chains have a bend to them, they're going to be more fluid or they're going to be liquid at room temperature. So these unsaturated fats, we usually see them as plant oils or fats. Our second lipid, the phospholipids, this also has a glycerol molecule. Attached to the glycerol you have two fatty acids, and then in that third spot you have something called a phosphate group. So this phosphate group is one of those functional groups that we looked at. So this phosphate group is hydrophilic, it's water-loving, whereas the two fatty acid tails are hydrophobic. So this is that amphipathic part of the phospholipid. So these phospholipids, because they're amphipathic, if you take some phospholipids, you throw them into water, they're going to orient themselves. So the phosphate head part, the orange circle, is going to stick out towards the water, whereas the fatty acid tails are going to stick in towards the middle, towards each other, because they're trying to get away from that water. So we'll look and see how these phospholipids actually create those biological membranes when we start looking at cells. The last type of lipid we're going to look at are the steroids or the hormones. Specifically, we're going to be looking at cholesterol. So cholesterol looks a little different compared to the phospholipids or the triglycerides. So cholesterol is actually 4 carbon rings hooked together. In cholesterol, we find that inserted into the phospholipid bilayer of our cell or plasma membrane. And like I mentioned, cholesterol is going to help regulate how fluid that membrane is going to be for the cell. Okay, so just to orient you, we're talking about our 4 different types of biological molecules. So we talked about carbohydrates, and then we just finished looking at the lipids. So our next one is going to be proteins, and then we'll look at nucleic acids. So the protein, the monomer or the building blocks are called amino acids, and there's 20 different amino acids that can be put together in lots of different combinations. So these amino acids, we join them together, they form the polymer, which is called a polypeptide. And these polypeptides, if you have more than one polypeptide interacting together, it's called a protein. These amino acids, when they're linked together, the bond that forms between them is called a peptide bond. Once all your amino acids are joined together, there can be hundreds, thousands of amino acids joined together in a string. Those amino acids are going to fold into a 3D shape. And I'll show you an example of this. Proteins have lots and lots of different functions, and I have just a few listed here. So they provide support for the cell. Some proteins work as enzymes, some proteins transport things. So hemoglobin in our red blood cells, that's a protein that transports oxygen. Other proteins are used for defense mechanisms, or they can be used for movement within the cell or for movement of the cell itself through its environment. Here are the 20 different amino acids, which are those monomers. These 20 different amino acids can be put together in lots of different combinations to help make proteins. So you can see that some amino acids are nonpolar, which means that they're hydrophobic, they're water-fearing. Other amino acids are polar, so they're hydrophilic, they are water-loving. And then other amino acids can be electrically charged, so they have negative and positive charges to them. So we're going to take these amino acids and we're going to hook them together to make our protein. So this is how we form this peptide bond. So you take 2 amino acids, they bond together through a peptide bond, And then you just keep on adding more amino acids to the chain, and this is going to eventually form into your protein. So protein structure, there's 4 different levels to protein structure. So your sequence of amino acids just in a straight chain, this is called your primary structure. Once you have all your amino acids hooked together, they're going to start to interact with each other. This is when you get to the secondary structure. So secondary structure includes the alpha helices, which you can see in the image. So they're kind of like a spiral shape. You also get beta pleated sheets, and this looks like an accordion or like a paper fan that you fold yourself. So this is your secondary structure. Once you have that, you'll have their tertiary structure. So this is when you get other types of interactions like a sulfide bond, for example. So this is when you really start to get that 3D shape. Some proteins are done once they get to the tertiary structure. They're ready to go. They're ready to do whatever their function is. Other proteins, you have to take more than one of these polypeptides. They're going to interact with each other, and this is called the quaternary structure. So an example of quaternary structure, if we go back to hemoglobin, hemoglobin's made up of 4 different polypeptides that are interacting together. And then that makes up your whole protein. Our 4th type of biological molecule we're going to be looking at is called the nucleic acid. So nucleic acids include DNA and RNA. I'm also going to show you ATP at the very end. So DNA and RNA, the monomer for these molecules is called a nucleotide. And a nucleotide consists of a phosphate group. You have a pentose or 5-carbon sugar, and then you're also going to have a nitrogen base. So those 3 things make up one nucleotide. DNA nucleotides, for their pentose sugar, they have a sugar called deoxyribose. So that's the yellow sugar up at the top. And then to that deoxyribose sugar, you have a phosphate group attached. And then you have one of 4 nitrogen bases. For DNA, you can either have cytosine, guanine, adenine, or thymine as your nitrogen base. So those are the 4 choices. An RNA nucleotide, instead of deoxyribose sugar, you have a ribose sugar, which is shown in red up at the top. And then for the 4 different nitrogen bases, you can either have cytosine, you can have guanine, adenine, or the 4th one you can have uracil. So DNA and RNA nucleotides are very similar to each other, but make sure that you can spot the differences. So they have different sugars, and then DNA has thymine, whereas RNA, it has a uracil nitrogen base. So in DNA, we take these different nucleotides, we hook them together to make a DNA strand, and then usually DNA, it's in the form of a double-stranded helix. So you actually have 2 strands that will hydrogen bond to each other to form a double helix. RNA, on the other hand, RNA is usually found as a single strand, so it's just a straight chain of these RNA nucleotides. So DNA forms this double helix, so you have 2 long polynucleotide strands, or single strands, and they're linked together by hydrogen bonds. And those hydrogen bonds occur between your different nitrogen bases on the nucleotide. And there's a few rules to how these 2 bases hook together. So thymine always binds with adenine. So you can see that at the very bottom of the image. Thymine and adenine will hydrogen bond to each other with 2 hydrogen bonds. Whereas guanine and cytosine, they always pair with each other, and they go together with 3 hydrogen bonds. So just remember, we have this complementary base pairing, or they're called Chargaff's rules. So adenine goes with thymine, cytosine goes with guanine. So this DNA. DNA is responsible for holding on to genetic information. So DNA has to be replicated when cells go through cell division. So DNA has to pass on this genetic information, this genetic message from one generation to the next. So DNA, it goes through replication, so each strand is copied. And replication is guided by that base pairing. So remember, adenine goes with thymine, guanine goes with cytosine. So at the very end of DNA replication, you get two separate double strands. So if you look at the image on this slide, you start with one DNA double strand up at the top. Those two strands separate from each other. You have complementary base pairing, and you end up with two double strands at the very end of this replication. The final part of nucleic acids is we're going to look at ATP, which is an energy molecule of cells. And we'll see this come up in other units during this course. So ATP is called adenosine triphosphate, so ATP. And adenosine triphosphate is actually a nucleotide. It has an adenine nitrogen base attached to a ribose sugar, and then instead of one phosphate, you have three phosphates attached in a line. These three phosphates, they hold lots and lots of energy in them. So the function of ATP is just to transfer and store energy for really short-term energy use. So we're going to be having cells making ATP during cellular respiration.